Hi, my name is Andrew Vizzerra, and I'm a member of Mike Barker's lab group at the University of Arizona. And today I'm pretty excited to talk to you about a cool story of biased fractionation in a homoploid and allopolyploid hybrid complex that we think is driven by the maternal parent. So we know that both polyploidization and hybridization are common evolutionary processes in vascular plants. Both of them are especially important in the evolution of allopolyploid species. But in the case of hybridization, it's interesting because there are inter and intralocus interactions between two different loci that came from two different parental genomes. And once they are joined in a single nucleus, there can be deleterious interactions given mutations and whatnot that cause genetic incompatibilities in hybrids. And that's especially the case in diploid hybrids. We often find them that they are sterile. However, following a polyploidization event, some hybrids can be fertile species. Now, following a polyploidization event, there can be a loss of genes at unequal rates. This is called biased fractionation. In the case of some genes being lost on one subgenome, but are still present on the other. And this bias can be persist through multiple rounds of duplications. And some work done in Arabidopsis, cotton, and the Brassicaceae have shown that genes in the more fractionated subgenome are more lowly expressed than those that are not. Which really leads us to the question of how hybridization and polyploidization really affect genome fractionation. And when we find some examples of biased fractionation in hybrids, we find a very small percentage of bias going on towards one parent. In the case of cotton, there's about a 3% bias towards one parental genome, whereas in coffee, it still falls within that range of about 1 to 3%. And this is a range that most plants fall into in hybrids biasing towards one parent. So it's not a very strong bias, but there is something going on there. In our case, we use a very special system called Selaginella. Selaginella are a really cool group of vascular plants because they're often coined as resurrection plants. Can basically come back from the dead given just a tad bit of moisture. And they come back with a vengeance as these large vegetative mats that grow on these rocky outcrops and slopes. But specifically, we're looking at two very closely related Selaginella species, Selaginella arizonica, which is found in a little bit more of Arizona, the Sonoran Desert, and Selaginella aramophylla, which we find in more Baja, California. Like I said, they're very closely related taxa, and they hybridize and produce very cool hybrids. And I say cool because they're so special because we have both a homoploid hybrid and an allopolyploid hybrid coming from the same parental cross. Now, these are relatively old hybrids. They're about 3 to 4 million years old. The Phoenix hybrid, I'm just going to refer to them from where they're found, as an allotetraploid, whereas the organ pipe hybrid, which we find in Organ Pipe National Monument, is our homoploid hybrid. And now, when we look at the ranges, we expect to find them in somewhere of a geographical intermediate between the two parents, and that's exactly what we find. However, they occupy more extreme environments. So this environment is about 1 to 2 degrees hotter and a little bit drier than the parental territories here. And this was some work done by Anthony Benyaga, and he found that both hybrids are morphologically intermediate of the two parents. But really what we need to address is if there's evidence for homoploid hybrid speciation. So we know that allopolyploid speciation occurred for the Phoenix hybrid, but we really need to see if homoploid hybrid speciation was occurring in the organ pipe hybrid. And that's a little bit difficult, but how we answered it was using a modified D statistic from Hibbins and Hahn. This statistic is looking at the difference in coalescence times and shows two models. So the first model The alternative model is one in which speciation is not occurring at the same time as hybridization. So if we look at the coalescence times, we find that maybe sometime in the past, this organ pipe hybrid diverged from the Arizonica parent. And then a little bit more recent in time, hybridized with the Eremophila parent, producing that signal that we see. So those two coalescence times will be different. Under the null model, however, it's expected that speciation and hybridization are happening at the same time. So those times of coalescence are going to be relatively similar. And what we do here is a simple comparison. We just subtract the two from one another. And in the alternative model, that D statistic is not going to equal 0, given the two different coalescence times. Whereas in the null model, that D statistic is going to be pretty darn close to 0. And when we perform this analysis, what we find is that there is evidence that hybridization and speciation are occurring at the same time, which is great. We have evidence that these are hybrid taxa of Selaginella arizonica and Eremophila, and that hybridization and speciation are occurring or occurred at the same time. So now my current work seeks to explore some of the genome architecture in these hybrids and parents. So what I did was a de novo genome assembly and annotation of these reference taxa, and then use some analyses like BLAST, reciprocal best BLAST hits, some Go analyses and phylogenetic analyses to further explore patterns of biased fractionation in these hybrids. So I just want to preface this by saying I'm working with very, very old short read Illumina data. So this is not going to be next generation sequencing type data, not going to be chromosome level assemblies. But basically what I did was use this relatively newer assembler called W2-RAP Contiger, which is a robust assembler that handles hexaploid genomes quite well. And what you could see here is that we find most of the genes, given the complete BOOSCOs found in these subgenomes. And we find that the Phoenix hybrid is definitely the largest, has the largest genome out of these 4 reference taxa. Now, since we don't have chromosome level assemblies and whatnot, we can't ask too specific questions, but what we can ask are content questions. So what are the parental contributions to each hybrid genome? And that's exactly what I'm going to explore. So after the assembly, I got these annotated using the breaker pipeline. And once they were annotated, I got these genes classified into cellular and molecular functions using the Gene Ontology Network and the Panther classification system. After this, I ran BLAST, a slight tweak of BLAST called the reciprocal best BLAST hits. What this does is just a one-to-one match in each hit. So we remove any sort of bias that might be driven by duplicate hits. So we only get a one-to-one match going on the forward direction and the reverse direction. And what we find is that there is a huge bias going on in each of these hybrids. So in the homoploid hybrid, there's a strong bias. There's a 12% bias towards the Arizonica parent, whereas the Phoenix allotetraploid, there's a 16% bias in the opposite direction. So now we have a bias towards the Eremophila parent. Now this struck us by surprise because not only do we have this huge, like one of the first cases of this huge bias going on, but it's also in a reverse direction. So one of the questions that we had was who their maternal parents were. Do they have the same maternal parent? Do they have different maternal parents? So maybe whoever contributed the maternal content to the genomes was the one in which these hybrids were biasing towards. And to answer that question, I did another de novo assembly of the chloroplast genome in this time around, and then a reference guided assembly of the contigs using Ragout and Selaginella lepidophylla as a reference, and then did some alignments and fed it to IQ-tree. And what we find is that there is evidence that supports our hypothesis before. So that organ pipe hybrid, that homoploid hybrid, is sharing more sequence similarity in the plastid genome with the Arizonica parent, and the allotetraploid hybrid is also sharing more sequence similarity with the Eremophila parent. So now we have evidence that supports our hypothesis that this is in fact driven by the maternal parent. But really that raised a couple of other questions, like what's going on in these plastid genomes? Is there maybe selection going on these plastid genes that make them a little bit more incompatible with each other? So only one parent is contributing that maternal content and thus biasing towards that parent and the nuclear genome. Well, that led us to this Vizzerra database. So Vizzerra is a really cool assembly of all the nuclear or plastid, cytoplasmic, and nuclear interacting genes classified into where they're found. Is it plastid targeting? Is it mitochondrial targeting, and whatnot? So what I did was extracted all those cytonuclear interacting genes that were present in our annotation set, and then wanted to do some selection analyses. And I did that using the busted model of HiFi. So busted just is basically a gene-wide test for positive selection. So it's not site-specific. And this test was performed on about 50 genes. So we found about 50 of those genes that were present in our annotation set. And we found that 6 of those were under positive selection. Of those 6, 4 were under positive selection in the Arizonica branch specifically, and 2 of which were under positive selection on the Aromophila branch specifically. Now I'm only showing the results of 2 of those genes. I haven't had too much time to explore these results further, but I just wanted to show you some early results that there is in fact some form of selection going on these genes that might be driving these incompatibilities in the hybrids, which makes them biased towards one parent, in this case, the maternal parent, than the other. But really, there's some future work to do in these plastid genomes. But what I've shown you so far, our story so far, is that there is evidence of a homoploid and allopolyploid speciation event happening in this system. And once I perform those genome content analyses, we find that these genomes are dramatically biased in one direction towards that maternal parent, which is really one of the first cases of such a strong and reciprocal bias that we know of. But like I said, there's still some future work to do in those plastid genomes. And we also have some rad-seq data that we're waiting to analyze to get some population genetic information from these hybrids. And with that, I just want to thank you for joining me today. And again, I just want to thank you, all members of the Barker Lab group, for your tremendous help and support.